All right, so start of this unit, we started out talking about the cell theory. Okay, and the cell theory, okay, we talked about the 3 points of the cell theory. All right, is that something I could ask in, let's say, multiple choice question? Certainly, because it sets up perfectly for a which of the following is not part of the cell theory, as there's only 3 options. Okay, so points of the cell theory. Okay, first one. Okay, it's actually the second one, but that's all right. Okay, the basic functions part here. Cells carry out the basic functions of an organism. All organisms are made of cells. Okay, and third point. Where do cells come from, Ireland? Right, okay. Alright, so cells reproduce by cell division. Alright, those are the 3 points of the cell theory. We also talked about the 4 kind of really important people to do with the cell theory, okay, and that was Hooke, okay, the accidental tourist, okay, who discovered cells in a piece of cork while doing an experiment with lenses. Alright, Leeuwenhoek, I think I even spelt that right. Okay, Leeuwenhoek, the guy who was the first person to look at living cells, had the microscope that looked like a ping pong paddle. Okay, and then Schleiden and Schwann. Okay, and those are the 2 guys who actually proposed the cell theory. Okay, so they're kind of important in that regard. Okay, again, probably not something that would be in written response, okay, but probably something that would be in multiple choice. Now, can I ask you about the cell theory indirectly in a written response question? Yeah, I can. Okay, there could be questions that set up where part of your explanation of the answer could have to do with, you know, maybe all organisms being made of cells. Maybe it's a surface area to volume kind of question, and that might be important. Okay, or it could be any type of function question. Okay, cell function, cell transport, whatever, where cells carrying out the basic functions could also be an important part of your explanation. As a recognition thing, like it would be a multiple choice question. Who looked at this first? A, B, C, or D, right? Like it just you'd have to recognize, not necessarily memorize. Yeah. Okay. So yeah, just remember, guys, that yes, there could be an outright recall question about these 3 points, but they're also probably especially point number 2. Okay, we've talked about it how many times in this unit. Okay, being an important thing, right, for explaining other ideas. Okay, after that we talked about microscopes. Okay, and lab drawings. Okay, there could be a question or 2 about use of the microscope. I mean, it would probably again be multiple choice type of stuff. But those could be important. Be very, very easy for me to ask a question like, here's a picture of something you looked at through the microscope. Draw a proper lab diagram of this. All right, again, I'm not going to be marking you on your artistic ability. I'm going to be marking you on format. Okay, so remember that in a lab drawing, it is done in pencil. What should you bring to the exam? Certainly a pencil. You might want to bring a pen too, but a pencil would be important if you were asked to do this. Okay, so if this is a sheet of paper, your name and the date go here, right? Your drawing goes in the middle. Your labels all go to one side. That was one of the things people did wrong in their labs. Okay, had their labels kind of going everywhere. And then the title, the size, and the magnification down below. All right, now you have a sheet that has all of that information on it already. Might be worthwhile to study that a little bit, okay, as preparation. So certainly lab drawings would be important. Remember also, part of this would be estimating size. All right, so I'm looking at something through my high-powered lens. Okay, my high-power lens has a field of view of 400 micrometers. The thing I'm looking at is this big. Approximately, how big is that? Okay, here's what I would do. I would say, all right, it's kind of small. If this is 400 across, I'm just going to kind of estimate how many would fit across here. So I'm just going to kind of draw them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay, I was going to guess that 20 would fit across. I'm a little bit off. Okay, but I would have a pretty forgiving range of sizes that I would accept here. I was going to estimate that this thing was about 50 micrometers. Okay, ends up now it's looking like it's more about 65. Okay, so I would say my acceptable range probably would have been between 50 and 100. Okay, on a question like that. Everybody with me? All right, it's not a difficult calculation, it's just kind of an estimation. But your estimation has got to be close. It's got to be within my forgiving range, okay, of sizes. Joshua, question? Well, I did. It's kind of about the same. It was kind of a circle slash oval. Yeah, you are supposed to measure along the longest dimension. You're absolutely right. Okay? No, if I was going to do that, I would say you're looking at it on high power, and I would remind you. Yeah, I would say you're looking through a lens with the field of view is this big. Yeah, something like that. Okay? All right, so that would be an important thing. Now, where is that going to be? Written response or multiple choice? Probably in the written response. Okay? Are those pretty easy marks? Yeah, those are really easy marks. That's definitely one we want to have. All right, next thing we talked about were the modes of nutrition. Okay, what are the two modes of nutrition? Anybody remember? Omar? Chemoheterotroph is one. And what's the other? Okay, Omar, you know it. Photoautotroph, all right. Okay, I would know those words for sure. Okay, and here's the other thing I would know about them. The differences in the cell structure. All right, we know that cells that are, let's say, animal versus plant, chemoheterotroph versus photoautotroph, have a lot of similarities. Okay, they both have a membrane. They both have a nucleus. We got that in the cell anatomy lab. Okay, but what are the differences? Okay, the differences are that these ones have to have lysosomes because they have to be able to digest the food they ingest. Okay, photoautotrophs are going to have cell walls for support and chloroplasts and water vacuoles. Okay, chemoheterotrophs may also have centrioles. Okay, so remember, there's differences in cell structure that are related to how these cells get their energy. Okay, so it would be important to know not only what those differences are, but why those differences are present. Okay, that could be multiple choice and written response, because there could be some explanation involved in that. Okay, or some maybe real world application type of stuff there. All right, after that, parts of the cell. Expect a diagram of a cell on your exam. Okay, obviously it will not be like your quizzes where you're asked to identify 16 things on one diagram. There will probably only be 5. Okay, but since you don't know which 5 I'm going to ask about, you have to know them all and what they do. Okay, so you got to be able to name and give the function just like on the last 3 quizzes. Well, you don't know. And you won't know until the exam which ones I pick. Sometimes I'm picking really obscure ones, and sometimes I pick obvious ones. Well, again, you don't know which cell I'm going to put on there. I'm only, there's going to be a diagram, maybe there's going to be two diagrams, maybe I'll put both, okay? The thing is you don't know, so you got to know both, right? And you got to know all the parts. Okay, so name and give the functions, okay? Those functions just have to be kind of like point form explanations. I'm not looking for, like paragraph answers about how a Golgi apparatus works, right? If you write, like for a Golgi apparatus, responsible for exocytosis or responsible for processing wastes, that's good enough. You know what it does, okay? So don't spend a whole bunch of time filling in a box with incredibly small writing, okay? That's only worth 1 mark. Okay, like if it's only worth 1 mark, I'm probably only looking for kind of a point form explanation. Okay, now if on the other side of that you went Golgi, waste, the waste what? Okay, you gotta have a little bit more than that on there. Everyone kind of follow me on that? All right, and then after parts of the cell are kind of on the same day when we talked about the functions, we talked about cellular transport. There are quite a few marks on cellular transport. Okay, what are the big 3 methods of cellular transport? Nope. Osmosis is one. Diffusion is another. Jane? Active transport. All right, here's what I'm going to ask about these. I'm going to give you a real-world situation and ask you which cellular transport would be most applicable in that situation, or which one I may be describing. Okay, and then you'll have to explain. First off, identify which transport process it is, and then explain. All right, everyone follow me there. Okay, so for osmosis, the big identifier that I'm talking about osmosis would be water, right? If it's talking about water, okay, it's definitely osmosis, okay? Water going across a membrane would also be, okay? Remember that osmosis works across the membrane. It does not work within the cell. It's only how water crosses a membrane. All right, and what's osmosis trying to do? Balance what? Salts, right. Active or passive? Passive. All right, so these are the things we should know, okay, kind of point form, okay, things we should know about osmosis, or the identifying points of osmosis. All right, diffusion. Okay, so high to low, it should be spelled like that, high to low. All right, and where does that happen? In the cell, or anywhere, really, right? Happens even in the air, but we'll say intracellular. That means within the cell. Okay, what else do we know about it? What can it transport? No. Anything. Right? You can transport any material. That could be sugar, it could be salts, it could be anything. Okay? Anything will work by diffusion. So transports any solutes, we'll say. Passive or active? Passive. Right? That's kind of its selling point, is that it's free. Okay, what kind of things might affect it? Riley? You can't control it? Okay, it's not targeted? Right, the larger the distance it has to travel, the less effective it's going to be. Surface area to volume is the thing that affects diffusion the most. All right, that could be a separate question, okay, about surface area to volume, but certainly diffusion has the biggest issue with that. Okay, and then active transport is obviously active, requires energy. Okay, what is it doing? Okay, Riley? No, osmosis moves water. Joshua? Right, against the gradients, okay? That means from low to high. Okay, so it is creating imbalance. Okay, creates imbalance, right? And is it within the cell or across the membrane? It's across the membrane, right? Okay, because remember, there were those special proteins that were in the membrane, okay, the transport proteins. Okay, they carry out this process. The diagrams that you had in your notes and that I showed you showed that the transport proteins essentially change their shape as they're pumping stuff across the membrane using active transport. All right, so I would say those are kind of the key ones there. Again, it's going to be, a cell needs to do the following. Which cell transport process would it use? Okay, that would be kind of maybe how I would word it. All right, a cell gets put into this situation. Which cellular transport process is going to happen? All right, and when I ask about cellular transport processes, I'm asking about these three things. All right, we did talk about endocytosis and exocytosis. Those are different. Okay, those are changes in the shape of the membrane that involve the enveloping and engulfing of material. These do not. Okay, these are cellular transport processes. The other two are the engulfing using the membrane. All right, so let's talk about the other two then. Okay, so endocytosis is taking stuff into the cell. Okay, taking into cell, and we had phagocytosis. Okay, and that had to do with food. All right, we had pinocytosis, which had to do with water. Okay, and we had receptor mediated. Okay, and that was for hormones. Okay, so that's, again, all three of those are mechanically the same. The cell's membrane engulfs that material and brings it in, right? Different from osmosis, from active transport, from diffusion. Mediated, that's an M, sorry. Okay, and then there was exocytosis. All exocytosis is the same. Okay, a vesicle, looks like a bubble, okay, containing waste or other products that need to be removed from the cell, goes to the membrane and the process reverses itself. Okay, so exocytosis is the reverse of endocytosis. Okay, those are not nearly as important as the big three. Okay, if I'm asking about these, they're probably multiple choice items. All right, whereas the big three would certainly be written. Response items. All right. Then related to that, okay, because after we got done talking about how cells transport things, we wanted to talk about, what am I on here, 6? Okay. We talked about cell size and transport. This was the cube activity that we did. All right, cell size and transport. Okay, has to do with surface area to volume ratios. All right, so you should definitely go over that activity, okay, as part of your studying. Okay, especially if you hopefully made notes on the stuff I talked about when I handed it back. Okay, those will be important things there. So surface area to volume ratios. How does that? Which cell transport process is most affected by this? Diffusion. All right, so that's something we need to remember. Okay, because you could have an apply your knowledge type of written response question. Okay, that might say, here's a situation where there's a very large cell. All right, or here's a situation where there's a very small cell, or here's 2 cells. How are they going to compare? All right, there's going to be something on the written response that's going to ask you about the surface area to volume ratio problem. Okay, as you get big, volume increases. Which one increases faster, volume or surface area? Volume, okay? And that slows down diffusion, et cetera, et cetera. So certainly an important topic there. Oh no, there won't be any measuring, no. All right. Yeah, so that's, again, another important concept there. Okay, so make sure you go over that one. And then we started on multicellularity. Why was multicellularity such a big advantage over being unicellular? Evan? Okay. Specialization, right. That was a big part of it. Okay, you have to have this if you're going to be a true multicellular organism. Right, that means you have cells or groups of cells called tissue that do a certain job. Okay, they're specialized to do that job. There may be other jobs that they don't do very well now or are completely unable to do. That's okay. They are interdependent. On the other cells, or with the other cells in that organism. Okay? What else did multicellularity allow an organism to do that a unicellular organism couldn't? More? Yeah, it has something to do with grow. Right, get bigger. Remember, we had the multi-micro, okay? In that cube lab, we had multi-micro. It was filled up with all the little micros. Doesn't matter how big you make that one. It's always going to fill up with little cells. And each one of those little cells was still going to have a good surface area to volume ratio. Being multicellular allowed them to overcome that problem. Okay? Right? Overcame this problem because they were made up of cells that were small, and because they were made up of interdependent, specialized small cells, they were able to get big. Okay? And bigger was better. Right? So multicellularity, kind of those ideas there. Maybe multiple choice, maybe written response. Okay, I'm still toying with that one a little bit. Right there. So understand kind of these relationships here. There may be an explanation part, or maybe I just figure out a couple of ways to ask that in the multiple choice. Okay. Again, I haven't really decided on that. All right. And then we talked about transport in plants. Okay, incidentally, we are going to look at our plants tomorrow. There are only a few of them that are showing significant movement of the dye so far. Okay, so we'll give them at least another day before we go and look at them. Right, so we'll actually have to bring them into this classroom tomorrow to look at them. Right, but that's no problem. Just know I haven't forgotten about that. It's just there isn't enough movement yet for us to get good observations. Okay, so transport in plants. What process is most involved there? Sorry, that's one of the factors. Which, yeah, osmosis is the transport one, right? Osmosis is what draws the water up the plant, okay? As the water evaporates, the salts gets left behind, blah, blah, blah. You need to understand that process very, very well, because I will ask you to explain it. I will ask you to explain it, which means it's going to be where? In the written response, okay? There's probably also some questions in the multiple choice about it. It's that important, okay? Now, water is obviously not the only thing that gets transported by plants. What else? Sugar, okay? So should we know about these two things? Okay, xylem is where the water gets transported, and that's on the inside of the stem. Phloem's on the outside of the stem where the sugar is transported, okay? And sugar is transported by what force? Gravity, right? Okay. So little things to remember there. Be able to explain that process really well. Look at that diagram that I gave you in your notes there, okay, and the numbered list of the processes. Okay, make sure that you can explain how those all work. And as we talked about transport in plants, we talked about the different parts and different tissues of the plant as part of this specialization thing. Okay, specialized tissue within the plants, we talked about roots, okay, and what their job is, right? We talked about the stem, right, and specifically with the stem we talked about those two things, okay, the phloem and the xylem, right, and how they work, and then we talked about the leaves. The leaves were the area where the most evidence of specialization was, okay, because within the leaf there were many different types of cells that were all specialized to do a certain job. Okay, so should you know that diagram of the leaf? Yeah, okay, so you probably should know about the epidermis, okay, and that it's responsible for this thing, right? The palisade layer, and what it does, the spongy layer, okay, and what it does, and these things. Okay, should also know about the stomates, made up of 2 guard cells, and how that works. Okay, because related to transport in plants was also, okay, conservation may not be the right word here, but okay, how a plant manages its water. All right, how it controls how much is lost by evaporation. What structures within the plant, okay, help this to happen. All right, so obviously the stomates are a really big part of that. Okay, what other thing on here is important for preventing evaporation? Well, spongy layer a little bit because that's where the water is, but one other structure that covers the plant. That one? Okay, cuticle, right? That's secreted by the epidermis. It's waxy. Okay, it seals the plant so water can't evaporate. Right? Those kind of things you should definitely know about H2O conservation, or management might be a better word. Okay, how does the plant manage its water? Okay, what are the structures and processes and things like that that the plant does to manage its water? I got to keep going here. Okay, it's all on the podcast. You'll be able to watch it later. Okay, so next thing here, guys, we talked about. I can't reach down here. My back hurts. Okay. Okay, plant growth. Okay, so we're looking at factors. Okay, factors that affect plant growth. We talked about that yesterday. Okay, what are some of those factors? How do they affect the growth of a plant? All right, sorry, it was a rhetorical question. Okay, that's something you guys need to go over. Okay, looking at, the things that we're going to talk about even more tomorrow when you do your lab design activity. Okay, so water and light and all that kind of stuff. Okay, you got your abiotic, okay, and biotic ones that you should probably talk about there. I would say this is probably more of a multiple choice type of thing, identifying them or being able to identify the effects of a specific factor, okay, something like that. Okay, and we talked about development as well. Okay, so we talked about development and growth, and when we talked about that, we talked about plant hormones. Okay, so that would be auxins. Not really, it doesn't really matter. Auxins, cytokinins. Okay, oh, sorry guys, I gotta move this up. I just can't bend that way. Okay, cytokinins, gibberellins. Okay, we had our ethylene and abscisic acid. Okay, so you should probably know what those things do, okay, what their role is. There could be pictures related to that. Okay, so here's a picture of a plant. What process is going on, and by what process I might be talking about tropisms. And then you can tie tropisms in to these. Okay, which one of these is most important for tropisms? Right, because that one causes the cells to get longer, and that's how a plant can bend and change its shape. Okay, so certainly be able to explain these and identify. Okay, from a picture. Okay, so explain and identify from a picture. Right, there's a lot of pictures on this test. In fact, I always photocopy this one in color. Okay, just because they're that important. So expect that you might see real pictures, also diagrams, but real pictures depicting real situations, and you have to decide from those if a tropism is going on, what hormone might be responsible for what's going on in that picture, what cellular transport process is going on in that picture, okay, those kind of things. So lots of diagrams in both the multiple choice and written response. Okay, that's it. Okay, that's what's on there. All right, now obviously this test is different, okay, than your chem test. Right, your chem test was a lot of formulas and skills. This is a lot more of understanding and recall. Okay, it's not just memorize and regurgitate, although there is some of that certainly. Okay, that's kind of the nature of biology, but it is also very much understanding. Okay, I know this stuff and I understand that I can apply it to this situation. I understand how it works in this situation. Right? So, you know, when you read some of those questions, some of them are going to seem like, well, you know, Mr. Codere didn't teach me that directly. Well, you're right. In biology, I don't teach you everything directly. Sometimes I have to see if you can extrapolate. Okay, that is, apply what you know to a situation. Does everyone understand there? Okay, so know that if I ask you a question, you know the stuff you need to answer it. It's can you apply that stuff to that situation? That shows a very high level of understanding of those concepts. Okay, so yes, you need to memorize the concepts for sure, but you have to think about how could Mr. Codere ask about these, and that's the trick to studying for biology is, all right, what are some weird situations that I can think of that this might apply to? Or maybe you sit down in front of the computer and you Google, you know, something about auxins and see. You know, okay, now I can see how that might work with this or whatever. That can be part of your studying for bio as well. All right, questions on that? Okay, certainly there's the easy stuff that's going to be the memorization, the parts of the cell and what they do. Okay, if you know that stuff, there's marks to be had for sure with that on the test, so definitely know that. you know, the big three cellular transport processes. If you understand how they work, make sure you write how they work, because that would be part of your explanation. Okay, stuff like that. The things that you can memorize are certainly important and worth marks. All right. So for studying stuff, read your notes. Okay. Watch the podcast, especially of things like quizzes. Okay, especially if you didn't do well on a particular quiz. Okay, watch the podcast of that quiz, and you can do that by going into PowerSchool. Okay, if you go into PowerSchool and go on a quiz that you didn't do well on and click on it, there'll be links below that have the quiz questions and the podcast of the quiz, so you can watch it again. Okay, always a good idea to do that. Look over your labs. Okay, and yeah, and then these two things here that I'm about to give you, okay, the review package and the pre-test thing that's got old exam questions on it. Evan, question? Yes. Any other questions there, guys? Riley? Yes. All right. Okay, so your first sheet there that you got, okay, is basically got your major concepts and minor concepts. If it's a major concept, it's something you should know really well because it's probably in both the multiple choice and the written response, or represents a significant number of marks on the exam, okay, and require kind of more high-level thinking. The minor concepts are more the kind of memorization stuff. Okay, they're still important to know. Something I forgot here, that prokaryotes versus eukaryotes, okay, which is number 6 in the minor concepts. Make sure you understand how they're different. Okay, we talked about that. Eukaryotic cells are the ones that have all the organelles, okay, where prokaryotic cells do not, and they're generally smaller, right? And then you've got a cell to label there. All right, so a diagram of that, okay, and then the chart for filling that in is on the other side. Okay, number 1, okay, is that kind of chemoheterotroph, photoautotroph type of question, okay, that we talked about in the review there. Okay, explain how plant hormones could be used to destroy plants rather than benefit them. So these are some kind of just here's topics that I'm going to talk about. Here's, you know, review them by answering these questions, kind of a thing. They're not necessarily exam questions, but they're kind of like that, right? And then the terminology, you certainly need to know the definitions, okay, for these types of things here. And then one's asking you here, draw a proper lab diagram. Obviously, we said that's likely to be on the test as well, okay? And then you've got your old exam questions there, okay, in that package. There's no key for this on my website, okay? This is just like the last time. If you want to find them out, you'll have to look them up. That's the idea. That's how you study, okay? But that's stuff I've asked on exams in the past, so it gives you a pretty good idea of level of difficulty and topics and stuff like that, okay? All right, so you've got about 40 minutes or so where you can work on getting your notes organized, okay, or you can work on these 2 sheets that I just gave you, okay. You've got some options there for things to do, okay. Questions? All right, have at her.